This is the first session, and I feel like it was a good opportunity to give a talk about how the idea of the R4 biotech came across, and we organized this concept of synthetic Rafts in bacteria. Initially, the whole idea came when we were in my lab, when we were studying signaling. My lab, at the beginning, we traditionally were interested in studying how cells interact with each other. And we were using for that a model organism. It's a bacterium. It's called Bacillus subtilis. It's used for molecular biology because it's really, really tractable. You can do genetic really, really fast, really well. It's a big bacterium, so you go to the microscope and it looks really well. So it has everything that you might dream to do molecular biology with bacteria. But in addition, it's a soil bacterium. So it also shows a very complex interaction play and signaling networks to detect neighbors or competitors in the niche. So it's also a fantastic model for signaling. So we were studying how Bacillus subtilis attach to roots or sense others, and we found this molecule, surfactant. It's a small molecule that Bacillus subtilis produces by itself. So it's kind of a quorum sensing. It tells to the bacterium how many of those are present in some niche by measuring the concentration of the molecule, and that triggers a response, which in this case is to form these biofilms, these pellicles. There are all the cells stick together and they form a matrix, so they are really strong and they protect themselves from antibiotics or other insults that are in the environment. So surfactant, just by concentrating the molecule in the medium, triggers this behavior, triggers this biofilm formation in Bacillus subtilis. So we were actually interested in biofilm formation and how this signaling forms biofilms in Bacillus subtilis. This is the normal assay that we have. This is the wild type. Forms this. It's a 24-well plate where you're seeing from the top. So you see all the cells making a biofilm on the top of the well. We randomly just generate a mutant, GSP, that didn't make the biofilm. Normally, the mutants that we do to study biofilm formation, they are somehow associated with the pathway. And here's the pathway for biofilm formation. There is a sensor. Man, I will see it later. Sorry, what? Oh. This is the sensor kinase that senses the signal. Surfactant is associated with the membrane, and that activates a phosphorylator regulator, SPO0A, and that triggers a whole and very complicated pathway to biofilm formation that ends up producing the two set of proteins that are necessary for matrix production. This is a structural protein in the matrix, and this is the polysaccharide matrix. It's a sugar and a protein that makes the whole biofilm. But for today, the important thing is that this sensor kinase can see activate this this regulator, SPO0A, and once you are here, the whole pathway gets activated. As I said, we accidentally got a mutant in which the pathway was not working. At least Bacillus subtilis cannot make the biofilm in these conditions. And there was two ways to restore biofilm formation in this mutant. One, of course, if we complement it with a wild-type copy of GSP, and then we have the biofilm. And the second option was to complement it with a version of SPO0A that is always active. It's fully active, so it doesn't need to be phosphorylated by kind C to be active. In this case, we see biofilm formation again. So that indicates that the defect in biofilm formation in this particular phenotype is associated with the kind C. The kinase is not working. You see, when we have this result, we start thinking about the idea of, well, what is this GSP doing? Why in this particular mutant the kind C is not functional? So GSP, this is what it does. It takes it looks like an esqualene synthase. It looked like an esqualene synthase. So it takes 2 farnesyl pyrophosphates and join them together to produce a molecule on squalene. So we purify the enzyme. We test the enzymatic activity by, we couldn't measure squalene, but we measure in the presence of farnesyl pyrophosphate how much phosphate is released, so the squalene can be formed. So, and we saw that it releases phosphate, so say, okay, it's an squalene synthase. So it is a molecule that produces some lipids, and somehow that affects the activity of kind C. We start thinking like these lipids are very unusual. Kind C, we know, is a membrane protein, so there should be some regions in which, or some areas in bacterial membrane in which kind C concentrates, and they are actually protected or reinforced by this type of lipids. Later, in a further collaboration with Eric Oldfield in the United States, we realized that probably the most robust hypothesis that we have right now is probably GSP is not an squalene synthase, because what we did at the beginning is to detect release of phosphate via a colorimetric reaction. But we measure actually if there is squalene formed in an in vitro with GSP, they didn't see it. They didn't see squalene formation. So, but they see release of phosphate. So right now, the hypothesis that GSP is producing is farnesol. So it's actually dephosphorylating farnesyl pyrophosphate and produces farnesol. Those are the lipids so far that we think, or at least one family of the lipids that we think are associated with these visionary domains that we are seeing. We were playing with the idea that there might be some domains. So from this point, we start taking all the literature of lipid rafts in bacteria and adapt all these protocols to do in our Bacillus subtilis. This is the basic assay to do lipid raft purification in eukaryotic cells. You basically take the cells and you treat them with a mixture of non-ionic detergents. It's a very mild treatment. Those detergents are not very hard, so they destroy the membrane of the bacterium, except those regions that they contain lipid rafts or microdomains, because those regions they are packed and they are compact regions, so they resist detergent disgregation and remain larger fragments that we can later separate in a sucrose gradient. And that's what we did. We separate a detergent-resistant membrane fraction, which is supposed to be enriched in these microdomains, and a detergent-sensitive membrane fraction that is supposed to be the phospholipid fraction. Here we see a bunch of proteins associated, different proteins associated with these two fractions. We were pretty excited. We were even more excited when we saw that kind C is associated with the DRN fraction. It's like there. And the GSP mutant, you see the amount of proteins that we detected in the DRN fraction is not a lot. We have actually one here that didn't change. So we actually see that the protein content on the DRN fraction in the GSP mutant, it's compromised. So it looks like these farnesol, these lipids that they make these domains, actually they do affect kind C, because in this mutant, in the GSP mutant, we were not able to detect kind C. That's something that we didn't detect in the DRN fraction, but we didn't detect it in the DRN fraction, and we didn't detect it in the cytoplasm. So probably it's, if it's not in the right place, it's degraded because it's probably not stable. Well, anyways, I think that is probably the reason why Bacillus subtilis couldn't make biofilms at the first place in the GSP mutant. After this, of course, we do mass spectrometry to identify what are these proteins that we find associated with the DRN fraction. We start checking what are these proteins. We found many proteins associated with signaling. That's what we were initially thinking, that they were bacterial lipid rafts. But there were also other proteins associated with cell wall synthesis, quorum sensing, protein trafficking like ABC transporters. These lipid rafts in eukaryotes are described to be for signaling and also for trafficking. So we were finding some similarities, but the best similarity that we found is these two proteins, FLOI and FLOT, Those are two scaffold proteins. They are called Flotillins, which are also found in the eukaryotic lipid rafts. Those scaffolds, they help, they provide stability to the rafts and also tethers interacting partners. So they promote efficient interactions between partners in the rafts. We found fascinating that we have two different Flotillins, Flo A and Flo G, in these lipid rafts of Bacillus subtilis, and immediately we totally forgot about kind C at all, and then we focused totally on the discovery of these two Flotillins. Here's the two Flotillins. You see, this is Flo A with GFP. This Flo T. You see, we have two different pictures. An exponential, when cells are growing, they're growing exponentially, there is a lot of food, they're happy. And where it's stationary, there is no food, so cells are like starving. The differences between the two Flotillins, Flo A and Flo T, is like we see expression of Flo A in foci. You see the number of foci in the membrane. There's probably expression like all the time, normal, pretty standard. Whereas expression of Flo T, even though you can see some foci, they are. The expression is boosted at stationary phase. So it looks like the two Flotillins are not the same. There's not redundancy. They have different regulation, so they might have different functions. So the first thing that we did was just start exploring what is this regulation, how are they controlled. We start, as I said, Bacillus subtilis is a fantastic model organism to do genetics, so we start knocking out one by one all the genetic regulators that Bacillus subtilis has to see if we see some differences in the distribution pattern of Flo A and Flo G. For Flo A, we never saw differences, like mild, strong differences that at this point of the when we were so naive, we were seeing. But in Flo G, we really see that when we have a mutation in SPO0A, remember the regulator I talked about that activates as a stationary phase and triggers all these behaviors. The mutation in SPO0A also inhibits the formation of these foci, and we can complement it with a double mutant ABRB. This is the pathway actually that controls it. So it is really controlled in a signal independent manner. This is what we think right now. This is the status of what we think the regulation of the two Flotillins in Bacillus subtilis is. We think one Flotillin is Flo A. The one up there is constitutively expressed. We always have it there. The number of foci, they never change. The other one, Flo G, it changes because it's controlled by a signaling pathway. So we have more expression of Flo T when cells enter a stationary phase. It has to have consequences. There's got to be a reason why there are two Flotillins differentially expressed. So we went into detail to see what are these Flotillins at the structural level, what are the difference to, what are they doing. This is the two Flotillins, Flo A and Flo G. You see there is one transmembrane region, and this PHB domain, that is it's a domain that is in all Flotillins and related proteins that nobody knows what is the function of it. It's just there in Flotillins, in stomatins, in prohibitins, and in other proteins, HFLCK. But nobody knows what is the real function of these domains. Flo A has around 300 amino acids, 339, I think. Flo T is like 509 amino acids. The major differences are in the C-terminal. This one is larger, and the C-terminal is just different. The N-terminal part more or less is the same with one transmembrane region and the PHP domain. The C-terminal part has 4 repeats like glutamic alanine. You see Flo A and Flo G has the 4. The Flotillins of eukaryotic cells, they also have these repeats. They also have these EA repeats. And in the eukaryotic field, it has been reported that these EA repeats are important for protein-protein oligomerization. So the scaffold activity of Flotillin relies on the presence of these EA repeats. We also have these EA repeats, so it might be also involved in protein-protein oligomerization in bacteria. Something that is different at the structural level is the number of foci that makes the two flotillins. Here is flow A. We normally count around 13 foci per cell. Flow G makes like 6. You know, it's flow A makes double. Here's but they, of course, when we do colocalization microscopy, we see some colocalization. The problem is that the number of foci is different. So we never, you see, it opens a whole question of, we have a huge variety of lipid rafts or bacterial rafts with different composition of two different flotillins. I cannot tell you whether this one that has, for example, only flow A doesn't have flow T at all. Maybe it has a little bit, and this one is the other way around. So from this one and this one, of course, we have some colocalization, but there is a huge variety of different lipid rafts that they might have different functions. So we were thinking, okay, we might have specialized rafts or different flotillins that play different roles within these particular microdomains. Here's a super resolution microscopy pictures, the flow A and flow T using the MEOS. I know MEOS is not the amazing way to do it because it might form aggregates, but we also do super resolution microscopy with photoactivating M-cherry. It doesn't produce aggregates. It's just the pictures are not that nice. So I'm going to show these ones. And believe me, at least with the photoactivating M-cherry, we don't see aggregates. There are 13 foci here, you see, but the foci are smaller. The number of proteins that concentrate in these foci are smaller than flow G. Flow G, these foci are way bigger, but there's only 6 of them. So at the end, when we do the maths, counting the number of clusters, we see that flow A makes more, but flow T contains more or less. When we make an estimation of number of proteins per cell, it's more or less the same. So the cells, they produce more or less the same proteins, flow A and flow T. It's just that they are distributed differently. So do they interact? Do they recognize themselves? Are they specific flow A to interact with flow A, or because we are seeing colocalization, but that doesn't really mean that they are actually interacting to each other. This is a bacterial to hybrid assay that we check interaction between flow A and flow A and flow A and flow T. We see a strong interaction between them, between flow A with flow A and flow T with flow T. But there is also, you see, there is also a strong interaction between different flotillins, flow A and flow T. It's, there's quite a communication, there's quite a network going on between the interaction of these two flotillins. And since I said before, in the eukaryotic field, the C-terminal EA repeats are described to be important for this interaction. We were making knockouts. We were making different variants of these flotillins to see which variation in these EA repeats affects the interaction of them. Indeed, when we affect the EA repeats, 1, 2, in 3, we didn't see anything, neither in flow A or in flow T, but when we delete the EA4, and the same happened with flow T, we also see that minimal alterations in this sequence, in this glutamic alanine, changes dramatically the affinity. They do not interact anymore, except with the EA3. And I don't have any explanation for that. I don't know what's going on with EA3. But the rest of the EA repeats are, you touch it minimally, and interaction just doesn't happen anymore. This is 4 for the 2 of them, for flow A and flow T. When we localize it by super resolution microscopy, now we don't, well, we see some foci, but the number of, the protein gets more dispersed. So the interaction is tougher in these particular variants, even though the protein concentration cells, they produce the protein at the similar level. One experiment that my PhD student, all this work was made by one PhD student when I was in Würzburg, Joe Schneider. He has a brilliant idea, and one day he came and said, Danny, if this is right, if it's the C-terminal part what drives oligomerization, and we have flow A that makes 13 foci per cell, and we have flow T that makes 6 foci per cell, and it's all about the C-terminal part, let's switch it. Let's put the C-terminal part of Flo-T in Flo-A, and the C-terminal part of Flo-A on Flo-T, right? So now this is, we put here, this is Flo-A that has the C-terminal. We replaced this part for this part. Now this Flo-A that has the C-terminal part of Flo-T, you see the number of foci changes. Now they are, it looks like Flo-A. It looks like Flo-T. So they are bigger foci. This doesn't make 13 foci per cell anymore. They are bigger. The number is fewer. And the same happens with Flo-T. This is a version of Flo-T that we replaced the C-terminal. Now we put the C-terminal part of Flo-A. It doesn't look Flo-T anymore. Now it looks Flo-A, like the 13 foci per cell. There you have the distribution. So really changing the C-terminal part, really changing this C-terminal oligomerization domain affects the oligomerization pattern. So it looks like this interaction, this C-terminal region is the interaction point in which flotillins recognize themselves and probably recognize other proteins, because the role of these proteins is to be a scaffold proteins to interact. So after this, we start thinking, well, I mean, probably these two flotillins it seems like they are so specific in interaction and recognizing to themselves and to other proteins. Probably they do attract different interacting partners to the Rafts. Probably this C-terminal region, it's important for recognizing different players in these Raft proteins. We need to identify them, which proteins binds which flotillin, whether there is different in functionality associated with the bulk of proteins that bind Flo-A and the bulk of proteins that bind Flo-T. So what we did, we run a native gel. We identify all the oligomers that binds Flo-A and all the oligomers that bind Flo-T, and then we send them for mass spec, see which are the proteins associated with each one of the flotillins. You know, this slide took us a year to do it, because at the beginning we did it wrong. We did pull downs. We would pull down Flo-A and pull down Flo-T, but we didn't think right, because we pulled down Flo-A and of course Flo-T pulled down too. So we cannot tell which protein binds to who. And the same with Flo-T. We pulled down Flo-T and Flo-A was there. So we have 50 proteins and we have Flo-A and Flo-T. So we cannot tell partners, right? So we have to actually separate them, identify the band, one band that is major in Flo-A that is not in Flo-T, get the mass spec and analyze it. Long story short, I would say generally the proteins that are associated with Flo-A mostly are associated with cell wall maintenance or turnover or membrane architecture. We don't know yet if it's a question of the functionality of the flotillin or it's because it's a constitutive protein. So it's always there from the beginning, and therefore it has more chances to interact with this type of proteins. In contrast, Flo-T, which is a protein that comes later, a stationary phase, of course, we show that interacting partners of this flotillin are more associated with the stationary phase, like biofilm formation, production of antibiotics, production of siderophores. Again, we don't know if this is because they are specifically programmed, these proteins, to interact with this cargo, or it's just because they come just at the time that these proteins are produced. And of course, there's a bunch of proteins that we know, that we see they interact with the two of them, with Flo-A and Flo-T, and this is like a box. There is a lot of proteins associated. Here, I'm going to show you today about 4R and RACI. There are two sensor kinases. The sensor kinases, we love sensor kinases because initially we have the idea of lipid Ras, of these platforms as lipid Ras. So they promote signaling, they promote efficient oligomerization of the signaling. Now we think differently. But at the beginning, these years, our concept of these platforms was for signaling. So we choose kinases because we thought that flotillin helps in the oligomerization of these kinases. Even though working with kinases is not really easy, because we need to overexpress them to see them. The concentration of these proteins in the membrane is really low. So you see at least we can, when we overexpress them, we are able to see them a little bit and colocalize with Flotillin. And then the interaction assays with the bacterial 2 hybrid, we also see interaction with the with one, like 4R, for example, interacts with Flotillin, but not with Floti. And Resi interacts with Floti, but not with Flo-A. So it seems like a specific interaction, at least in this heterologous system. This is a bacterial, we call it bacterial 3 hybrid assay. It's a bacterium that produces Resi. So we will measure the interaction between these two proteins, oligomerization, let's say, dimerization of the kinase. And we are going to make different strains that produce a different concentration of Flotillin. In this case, we are going to have no concentration of Flotillin, no Flotillin at all, a little bit. So you have here a little bit produced, medium here, and high production of Flotillin. And then we compare these strains that they do the same interaction assay in the presence of different concentrations of Flotillin. So you see that when we increase the concentration of Flotillin, the interaction improves, except for this higher superproducing Flotillin strain, the interaction goes really long. And the same happens with 4R. So we have a mid-level until here we peak in the best interaction, and then for the maximum it just goes down. This is exactly how it is described in eukaryotes, the activity of a scaffold, this type of scaffold proteins. There is always a concentration of the scaffold that is optimal for the interaction to happen. So when you have a lot of the scaffold, it titrates the components, it titrates the interaction partners, and then interaction doesn't happen anymore. So it looks like these scaffold Flotillins that we identify in our platforms, they do really act as a scaffold in this cargo, in these signaling kinases to promote the interaction. Of course, we do microarray to verify that the mutants in 4R are similar, at least for this particular regulon, that the mutants in Flo-A and mutants in Flo-T are similar for these particular pathways that the mutants in Flo-T. This is only 2 genes that we selected from the whole pool of genes that are controlled. These ones are controlled by 4R, which are associated with cell wall turnover, and these ones that are controlled with Flo-T, is production of antibiotics. As I said, Flo-T is more associated with antibiotic production and secondary metabolism, and Flo-R is more like primary metabolism. So it looks like the absence of the Flotillins affect some of the genes that control the kinases, which is expectable because we know that in the absence of the kinases, the absence of the Flotillins, the kinases are not oligomerizing at least as efficient as they should in the presence of Flotillin. I had to be honest with you, the hardest part always in studying these Flotillins is the phenotypes. It's really hard because what they do is they promote efficient interactions. So the phenotypes are, you can find a delay, or the reaction doesn't happen so efficient, so it doesn't produce that much this, but there is no like a clear, it's not one of those phenotypes that they find in the 80s, right? There is no flagella and it doesn't move around. I don't think these phenotypes are there anymore. They're all published. So in this particular case, it's hard. We were actually checking if there is any associated defect that we can exclude from associated with one Flotillin or the other. We find great phenotypes in the double mutant, but the single mutants, until this work, we were not able to see anything. But once we know that where to look for, we know that, I mean, probably Flo-A is associated with cell wall, Flo-T is associated with stain with the Resi, and this sensor kinase is associated with stationary phase. So let's go there. Let's go to find some phenotypes. I'm not saying that they are great, but we found some. So in this case, in this particular medium, you see the culture of Flo-A mutant and only Flo-A mutant gets pink. And gets pink because it produces some amino acids that dimerize and chelates iron. And it forms an artifact that is pink, and it's called butyramine. In the literature, it says that it has no function in Bacillus. It's just artifactual produced. We think this is consequence of releasing amino acids to the medium because they cannot use it for cell wall synthesis, because Flo-A we know that it's associated with cell wall, and they need these amino acids for the cell wall. But that's we think. We don't have any more proof than that. And actually, I'm very happy to see pink. For this one is more robust because this REST pathway activates in oxygen-limiting conditions. So if you have some defect, when you have a reduced oxygen, it doesn't grow that well as the wild type. So this one really matches very well with the interactions that we have seen in the microarray. So this is the last slide to show you what we think right now. We have two flotillins in Bacillus subtilis. They attract different interaction partners. One is constitutively expressed. The other one is activated by quorum sensing. We know the signatures that brings proteins to the Rafts. With this idea, we decided, we can do synthetic Rafts. We can actually take this knowledge and put enzymes that they have problems to interact, and in industry generates like very expensive products. We can try to put them together in the Rafts to see if the Rafts help them to interact, and we can make it cheaper or faster, so we can improve efficiency in industrial reactions. And this is the whole idea of R4 Biotech. I actually made up this logo, and I'm really proud of it. And Jean-Marc Gigot told me that I have no taste, so they put this one there. And
no one's alive.
Yeah. So, and I think this is my last slide, so thank you very much for your attention, and I'll take any questions.